Cek. Gue panggil dulu.
Katanya mau ini dulu, tarik dulu.
Ya dibaca dulu.
Sini aja nggak apa-apa dulu. Sini dulu nggak apa-apa.
Berapa menit? Ladies and gentlemen, we present to you a traditional dance, Sigeh Pangunten from Lampung, Indonesia. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Tabi pun. Good morning, ladies and gentlemen. Welcome to Meeting Room Politeknik Negeri Lampung. We proudly present the second International Conference of Agriculture and Applied Science 2021. It's being a great honor for me, Marlinda Apriani, to be standing here and welcome each and every one of you in the second International Conference of Agriculture and Applied Science 2021 with themes Innovation and Application of Sustainable Agricultural Technology as a Solution for Food Security. Ladies and gentlemen, over the next few hours, this meeting will provide you plenty of opportunities to discuss, share knowledge and insight. Before we proceed the programs today, we would like to express our sincere gratitude to Excellency, the Director of Politeknik Negeri Lampung, Dr. Insinyur Sarono, Magister Science, Excellency, Professor Bunyamin Taran, PhD, from University of Saskatchewan, Canada. Good night, Prof. Excellency, Professor Dr. Rosma Ahmad, from University Sains Malaysia, Penang, Malaysia. Good morning, Prof. Excellency, Ruekada, PhD, from Shijonawate Gakuin University, Osaka, Japan. Good morning, sir. Excellency, Mr. Anung Wahyudi, PhD, from Politeknik Negeri Lampung, Indonesia. Good morning, sir. And Excellency, distinguished guest delegates, ladies and gentlemen. Excellency, our distinguished guests, ladies and gentlemen, to increase the spirit of our nationalism, let us sing the national anthem, Indonesia Raya. Audience, please rise. Thank you. For our program today to run smoothly, let's us pray to Allah Subhanahu wa Ta'ala. Prayer reading will be led by Mr. Studio, MPD.
Warahmatullahi wabarakatuh. Ladies and gentlemen, let's pray together for a moment to Allah subhanahu wa ta'ala. Allow me to lead a prayer in Islam For the other religious, please adjust. إلى هذه وإلى كل نية صالحة. الفاتحة. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين. إياك نعبد وإياك نستعين. اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين. الحمد لله رب العالمين حمدا شاكرين حمدا ناعمين حمدا يوافي نعمه ويكافئ مزيده. يا ربنا لك الحمد ولك الشكر كما ينبغي لجلال وجهك الكريم ولعظيم سلطانك. اللهم صل وسلم وبارك على سيدنا محمد وعلى آل سيدنا محمد كما صليت وسلمت وباركت على سيدنا إبراهيم وعلى آل سيدنا إبراهيم في العالمين إنك حميد مجيد. اللهم لا سهل إلا ما جعلته سهلا وأنت تجعل الحزن إذا شئت سهلا. اللهم إنا نسألك علما نافعا ورزقا واسعا وعملا متقبلا. اللهم إنا نسألك الثبات في الأمر والعزيمة على الرشد ونسألك سكر نعمتك وحسن عبادتك ونسألك قلبا سليما ولسانا صادقا ونسألك من خير ما تعلم ونعوذ بك من شر ما تعلم. اللهم انفعنا بما علمتنا وعلمنا ما ينفعنا وارزقنا علما تنفعنا به. اللهم إنا نسألك الهدى والتقى والعفاف والغنى. ربنا آتنا من لدنك رحمة وهيئ لنا من أمرنا رشدا. اللهم اجعل جمعنا هذا جمعا مرحوما وتفرقنا من بعده تفرقا معصوما برحمتك يا أرحم الراحمين. ربنا لا تزغ قلوبنا بعد إذ هديتنا وهب لنا من لدنك رحمة إنك أنت الوهاب. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار. وصلى الله على سيد النبي الأمي وعلى آله وصحبه وسلم. سبحان ربك رب العزة عما يصفون وسلام على المرسلين والحمد لله رب العالمين. آمين آمين يا رب العالمين. والسلام عليكم ورحمة الله وبركاته.
Excellencies, our distinguished guests, ladies and gentlemen, without further ado, we would like to invite to the podium to deliver his welcoming speech as a Chief of Research and Community Development Polytechnic Negeri Lampung, Mr. Dr. Yana Sukaryana, Magister Science.
Assalamualaikum warahmatullahi wabarakatuh. The Honorable Director of Polytechnic Negeri Lampung, the Honorable Assistant Director of Academic Affairs, Administration and Student Affairs of Polytechnic Negeri Lampung, the Honorable Head of Unit and Section at Polytechnic Negeri Lampung, the Honorable Professor Bunyamin Taran, PhD, University of Saskatchewan, Canada, the Honorable Professor Dr. Rosma Ahmad, University Science Malaysia, the Honorable Professor Rohai Kada, PhD, Shijo Nawate, Gakuen University, Japan, the Honorable Anung Wahyudi, PhD, at Polytechnic Negeri Lampung, Indonesia. Welcome to the International Conference on Agriculture and Applied Science 2021. The second international conference held by Polytechnic Negeri Lampung. International Conference on Agriculture and Applied Science be held on 18 November 2021 in Polytechnic Negeri Lampung, Indonesia. This day will be dedicated to cover a wide spectrum of them related to innovation and application of sustainable agriculture technology as a party for participating in preparation of International Conference on Agriculture and Applied Science. The Honorable keynote speaker, and also thank you for your willingness to be a resource person in International Conference on Agriculture and Applied Science. We hope this conference will be of professional and personal benefit to all of you as we process with our work toward a future food and agriculture situation that is sustainable and capable of providing food security and good nutrition for all. We appreciate for your participation and invite all participants to return to join the next ICOAS. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you, Mr. Dr. Yana Sukaryana, Magister Science, for the welcoming speech. Ladies and gentlemen, it is now my deep pleasure to introduce the Director of Polytechnic Negeri Lampung, His Excellencies Dr. Insinyur Sarono, Magister Science, to deliver opening speech, and afterward, we would like to request the Director to officially open the second International Conference of Agriculture and Applied Science 2021.
Assalamualaikum warahmatullahi wabarakatuh. Good morning and tabi pun. Alhamdulillahirabbil alamin. Wassalatu wassalamu ala asrofil anbiya'i wa mursalin Sayyidina wa maulana Muhammadin wa ala alihi wa sahbihi ajma'in, amma ba'du. Firstly, let's thank and pray unto my God, Allah subhanahu wa ta'ala, who has given us some blessing and mercies, so now we can meet and gather in this meeting without any troubles. Secondly, may salam and salawat always be given to Prophet Muhammad sallallahu alaihi wasallam, who has guided us from darkness to brightness. Dear ladies and gentlemen, welcome to the International Conference on Agriculture and Applied Science ICOAS 2021, the second international conference held by Polytechnic Negeri Lampung. Polytechnic Negeri Lampung is a vocational university focusing on developing student applied skill and conducting applied research. We hope that applied research can contribute to prosperity of the society. The ICOAS 2021 is expected to accelerate the dissemination of the result of the applied research. I hope this conference can become an arena for discussion and collaboration regarding the latest research topic. Dear ladies and gentlemen, this conference would have not been possible without the contribution and hard work of Cannot Speak, Invited Speak, all the author and reviewer and advisory committee as well as chairperson. Allow me to take this opportunity to express my sincere appreciation to all of you. Due to COVID-19 pandemic and following the advice of local authority, the conference will be held through online or virtual conference. As the Director of Polytechnic Negeri Lampung, I hope I have the honor to welcome all plenary speaker. The first, Professor Bunyamin Taran from Department of Plant Science University of Saskatchewan, Canada. The second, Professor Dr. Rosma Ahmed from School of Industrial Technology, University Science, Malaysia. The third is Professor Roihyo Kada from Sionawate Gakuen University, Japan. And the fourth, Mr. Anung Wahyudi PhD from Plant Breeding Polytechnic Negeri Lampung. All Indonesian presenter and participant to ICOS 2021, sincerely I hope that you will enjoy on the conference. And thank you for your participation. Just this my speak. I do apologize to you all if I have a lot of mistake. Thank you and see you later, all of me. By saying Bismillahirrahmanirrahim, I opened the ICOS 2021 at Polytechnic Negeri Lampung. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Cek. Thank you for Dr. Sarono, Magister Science, for opening speech. Excellencies, ladies and gentlemen, that's our opening ceremony today. Thank you for your kind attention. Before we come to our main program, main session, we are now in a coffee break. Please be noted that break is only 5 minutes. Thank you. Yuk.
sejenak renungkan, kita diciptakan berbeda demi satu pesan yang sejuk untuk saling melengkapi. Memang tak banyak orang yang mengerti tentang ini, tapi ikatan batin kan menggema sepanjang waktu. Percayalah, kita melegenda di sana. Pilih masa depanmu di mana mimpimu. Jika kita pernah berbagi air mata, sahabat jangan lupa bahagia bersama. Percayalah, kita melegenda di sana. Pilih masa depanmu di warna mimpimu. Jika kita pernah berbagi air mata, sahabat jangan lupa bahagia bersama. Percayalah, kita melegenda di sana. Pilih masa depanmu di warna mimpimu. Jika kita pernah berbagi air mata, sahabat jangan lupa bahagia bersama. Sahabat jangan lupa bahagia bersama. Vokasi berjaya. Ada makna yang akan terungkap tentang perjalanan yang berat untuk kita ukir bersama. Jadi sebuah cerita pahit ketilnya kisah ini, namun kuat dan menguatkan sebagai perekat setiap hati yang condong pada mimpi. Memang tak banyak orang yang mengerti tentang ini, tapi ikatan batin kan menggema sepanjang waktu. Percayalah, kita melegenda di sana. Pilih masa depanmu di warna mimpimu. Jika kita pernah berbagi air mata, sahabat jangan lupa bahagia bersama. Baiknya sejenak renungkan, kita diciptakan berbeda demi satu pesan yang sejuk untuk saling melengkapi. Memang tak banyak orang yang mengerti tentang ini, tapi ikatan batin kan menggema sepanjang waktu. Percayalah, kita melegenda di sana. Jika kita pernah berbagi air mata, sahabat jangan lupa bahagia bersama. Percayalah kita melegenda di sana, pilih masa depanmu, juara mimpimu. Jika kita pernah berbagi air mata, sahabat jangan lupa bahagia bersama. Sahabat jangan lupa bahagia bersama. Vokasi berjaya.
Assalamualaikum warahmatullahi wabarakatuh. Salam vokasi, salam Free Factor Indonesia.
Perkenalkan, nama saya Yohanes Hendrianto serta rekan saya
Ahmad Farori Al-Hasani
dan rekan saya
Wulan Rahmawati.
kami dari program studi Sarjana Terapan D4 Teknologi Perbenihan Politeknik Negeri Lampung. Pada program Free Factor Indonesia 2021 kali ini, kami mahasiswa vokasi Sarjana Terapan Pertanian akan menampilkan karya kami yang berjudul Bertani ala Milenial menggunakan Bracket Pot berbasis Internet of Things sebagai solusi pertanian urban di masa pandemi. Inilah karya kami. Di masa pandemi COVID-19 yang belum tahu hingga kapan berakhirnya ini, pemerintah membatasi kegiatan warganya di luar rumah, dan kita mengenalnya dengan istilah work from home. Hal ini dimaksudkan untuk mencegah penyebaran virus COVID-19. Sebagai mahasiswa vokasi, kami melakukan inovasi pertanian ala kaum urban milenial. Sistem vertikultur, hidroponik, dan multiple cropping kami sederhanakan dalam sistem bertani di lahan terbatas menggunakan bracket pot berbasis IoT sebagai solusi urban farming masa kini. Apa itu bracket pot? Bracket pot adalah sistem rambatan tanaman yang menjalar seperti semangka dan melon menggunakan pipa paralon, kawat, benang, dan tentunya pot dengan media organik. Sistem bracket pot ini mudah dalam pembuatannya, efektif di lahan sempit atau terbatas, seperti pekarangan rumah. Bahan paralon yang awet atau tahan lama, dan menggunakan media organik yang mudah didapat, dan tentunya tanaman yang ditanam menyehatkan bagi tubuh kita. Tanaman yang kita tanam dan kembangkan benihnya yaitu kultivar melon oriental Macquarie yang memiliki nilai jual yang tinggi, unik, fresh, dan healthy fruit karena ditanam pada media organik. Sayuran seperti sawi, bayam, kangkung, pakcoy, selada, dan lain-lain menjadi tanaman multiple crop dalam sistem budidaya bertani ala milenial yang kami kembangkan. Inovasi ini telah kami ikutkan di Lomba Inovasi Pertanian tingkat nasional tahun 2020 yaitu Panah Merah Innovation Award atau PMIA 2020 dan mendapatkan juara 3 serta menjadi juara tim terfavorit mewakili mahasiswa vokasi Politeknik, sedangkan finalis lainnya dari mahasiswa universitas.
Selamat untuk juara 3 Yohanes Hendrianto, Watini Hefri Jayanti, Afrida Aruma Ningrum.
Program studi D4 Teknologi Perbenihan Politeknik Negeri Lampung memiliki teaching factory dengan nama Seed Teaching Farm atau disingkat STEFA, yang mana memberikan wadah bagi kami para mahasiswa untuk berkarya dan berinovasi. Sinergi kegiatan yang kami lakukan adalah pengembangan produksi benih dan buah melon oriental cultivar Macquarie, yaitu Ogan Macquarie dan Ginseng Macquarie. Buah ini memiliki nilai jual yang tinggi, namun mudah dalam budidayanya, serta waktu panen yang relatif singkat, yaitu 60-65 hari setelah tanam, atau sekitar 2 bulan saja sudah bisa dipanen. Produk yang kami hasilkan adalah benih F1 melon Macquarie, serta buah melon kultivar Augan dan gingseng. Produk lain dari multiple cropping adalah sayuran segar yang dipanen hanya dalam waktu sebulan saja. Sehingga selama penanaman melon Macquarie, kami mampu memanen sayuran tersebut sebanyak 2 kali. Tentunya hal ini akan menjadi sumber pendapatan masyarakat yang dihasilkan di pekarangan sendiri. Internet of Things yang kami terapkan untuk memantau data lingkungan pertumbuhan tanaman seperti suhu, kelembapan udara, kelembapan tanah, intensitas cahaya, relative humidity, pH tanah, dan lain-lain guna mengontrol lingkungan tumbuh tanaman. Sistem pengairan yang kami gunakan adalah sprinkle kabut yang kami kontrol menggunakan Android. Serta drip tetes memudahkan dalam memenuhi kebutuhan air dan nutrisi tanaman. Demikianlah yang dapat kami sampaikan dari karya kami. Tetap semangat dan tetap produktif di masa pandemi. Wassalamualaikum warahmatullahi wabarakatuh. Salam vokasi.
Excellencies, ladies and gentlemen, now we come to our main programs. Main session will be led by our moderator, Dr. Fitriani. First of all, allow me to introduce our moderator, Dr. Fitriani. Ms. Dr. Fitriani is an associate professor in agricultural economics, lecturing in food agribusiness study program, Polytechnic Negeri Lampung since 2000. She's received a grant and scholarships from Dissertation Grant LPDP in 2016 to 2017 and invited speaker in format Symposium Fukuoka, Japan in 2018, regional training program on developing and implementing agro-industrial strategies in at Chiang Mai, Thailand, AFMA FAO 2015. Research Exchange Grant, JASO, Japan. Now she is Editor-in-Chief Journal of Food System and Agribusiness, or JOPSA. Well, ladies and gentlemen, I'd like to invite Ms. Dr. Fitriani to lead discussion together with the keynote speaker. Please, Ms.
Bismillah. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you very much for Ms. Marlinda, who nicely introduced me in this prominently session for International Conference of Agriculture and Applied Science 2021, held by Polytechnic Negeri Lampung. It is honor me as a moderator to facilitate the wonderful idea in innovations and applications, sustainable agriculture, technology, solutions for food security. I delighted to inform that in these occasions we have 4 wonderful keynote speakers come from Asia and American regions. Firstly, I would like to introduce the prominent keynote speakers for the first sessions. Professor Bunyamin Taraan from University of Saskatchewan, Canada. Allow me to inform the curriculum vitae of Professor Bunyamin Taraan. Hello, Professor Bunyamin. Waalaikumsalam warahmatullahi wabarakatuh. It's so delighted to have you in this wonderful sessions. Yeah. Firstly, I met you at 2018 at University of Lampung on the class workshop writing international journals. Yeah, I think it's a great opportunity to me to meet you and advise, supervise, also supervise about how to good writing in international journals. Allow me first to introduce your curriculum vitae, Prof. Professor Buryamin Taraan, he's a graduate from bachelor from Sriwijaya University Palembang, Indonesia, and he continued to the master and the PhD from the University of Guelph, Canada, and finished at 1999. For the career, firstly, Professor Taraan starts from as lecturer in University of Sriwijaya, and then continue as a postdoc fellow in Biotechnology Center, Department of Genetics, University and Wisconsin's Medicines, and now he is in charge as Ministry of Agriculture Strategic Research Chair in chickpea and flax seeds breedings and genetics in Department's Plant of Science, Crop Development Center, College of Agriculture and Bioresources, University of Saskatchewan, Canada. I think the prominent paper from him is with title The Innovation and Application of Technology Sustainable Agriculture as a Food Security Solutions. The bottom line is, can we feed 10 million billion people without damaging the environmentals? Please, Prof. Taraan, the time is yours. Yes, clearly.
Wabarakatuh.
Waalaikumsalam warahmatullah.
First, I'd like to thank you. What we are doing, what we have done in Canada for everybody here. So I'd like to start my presentation. Please allow me to share. Okay, so we'll open. Can you see my slides? Can everyone see my slides?
Yes, it's clear.
Yes, okay, good. So my name is Buryamin Taraan. I'm a professor and also plant breeder at the Crop Development Center at the University of Saskatchewan, and especially working with two major crops, chickpea and flaxseed. Flax is also known as linseed. And also on a smaller scale, I'm working on the coriander breeding and genetics as well. So I was given the title for my presentation is about innovation and application of technology, sustainable agriculture as a food security solution. So this is the title given to me. So basically there are, okay, let me start. There have been many definitions of sustainability, strategy, and food production. Okay, so but the bottom line is that everybody knows that by 2050, it is estimated and will be reaching out 10 billion people globally. So the bottom lines in any talking about sustainability, food security is that can we provide the food for those 10 billion people without damaging the environment? So this is the essence of any talking, any strategy, any technology with sustainability and food production. Okay, how we can provide the food, nutritious food for those 10 billion without damaging the environment. Okay, so the challenge is not right there. So not only there, but there are people kind of other challenges as well. For example, the increasing global population, as we know, will be reaching 10 billion by 2050, which is about 30 years from now. It's not long. Climate change, increasing temperature, okay, and unpredictable climate pattern. Okay, so that's another kind of challenge that we have to work with when we're talking about sustainability in food production. The shortage of more potable water, okay, and as well as the shortage for the irrigation, the quality of water, that's another one. And then the loss of arable land due to urbanization and other purpose, so kind of refunctioning the land that used to be productive agriculture for different purpose. That's happening globally in many places in the world. So that's another challenge. So what left for agriculture now is more and more like some of the marginal land. Okay, so that's, and then the increasing food wastage. Food-related issues at the same time also happening, malnutrition, obesity. Okay, so one group have too much food and the other group do not have enough food. So as those are kind of adding to the challenge of reaching out the sustainable food production globally. So my talk is, as I'm a plant breeder, plant geneticist, I'll be focusing more into the plant breeding. What we have done in plant breeding as a technology, as a techniques, to develop plant varieties, because in any production, it started with the varieties. You have to have good genetics to be able to maximize your production. Okay, so in plant breeding, there are three major aspects that we can we try to achieve. The first one is stabilize and increase yield despite the changing climate. So in other words, how we increasing the yield potential of any crop despite the increasing temperature, the kind of unpredictable weather pattern, etc. Second is that how to develop varieties that can withstand pest and disease with fewer crop inputs. Okay, it's not much more use of fungicide, herbicide, or any chemical, but how we can develop such varieties. And then to maximize resource use efficiency, how to make the plants using kind of minimum amount of water, less water use, and less space, and less nutrient at the same time to maximize their production. So this is a big challenge. This is a big goal in plant breeding. And at the same time, we also been the inherent process in plant breeding is a long process. So, okay, so for example, just to improve one character in rice, for example, it still take about 5 to 7 years, or even sometimes up to 12 years. Okay, so in the conventional way of plant breeding, we include kind of many generation of crossing and selection. Okay, and at the same time, there's a concern about narrowing the genetic diversity, linkage drag, and labor intensive. So this is the challenge that everybody is facing when you're working with plant breeding. Okay, so nowadays, with the advance of the genetics and breeding, so in genomics, so modern plant breeding, we use all kind of techniques available. We use molecular technique, including DNA sequencing, RNA sequencing, proteomics, phenomics, putting all together to make the process really precise and can be done in much shorter ways than conventional breeding. So we do still need field assessment, like you see on the picture on the corner there on the far left, where I was kind of assessing some of the linseed breeding lines. Okay, so the final still need to be done in the field to make sure that the varieties will produce as you expect. So for those things in genomic, we already have some resources. So this is the crop as I'm a plant breeding with the legume. So I involved in mostly in legume crops. So this is the resources that we developed in legume that were published already a few years ago in chickpeas, in Nature Biotech 2013, and wild chickpeas in Nature Communication, and recently kacang ercis in Indonesia, also known as or field peas in Nature Genetics. So this is all the resources are available online. Okay, so I mean available to the public, so you can access all the information here. Okay, so one application of such information is that we do kind of a lot of things so-called genome-wide association analysis, where you kind of matching the variability within the genetic materials of the population and also their phenotypic expression, and then to know which of that genetic material or DNA sequence or part of the gene that are associated with, for example, yield or taste or sugar content, you name it, in any crop that you're working with, protein content. So in other words, we do like gene discovery with this analysis, as well as different population structure. You can use biparental population for QTL analysis. So this has been done already. Okay, so I can summarize here, for example, this is the major QTL, or if you have a chickpea, there are 8 chromosomes. So we know the location of the genes or part of that chromosome that control the various abiotic stress in chickpeas. This is already published in Theoretical Applied Genetics in 2020. So we know the location of the genes. So the application for this is that instead of doing the phenotypic section, for example, screening plants under the drought, we select this directly using the DNA variability itself. Okay, so when we pass the verification, so we use that information in making the selections. So instead of growing plants under certain conditions, we can cut that process by selecting at the early stage of plant growth by using a DNA profile. So the other, this is kind of quite new. This is started with animal breeding, okay, so by in 2000. So this is what we call genome-wide genome selection. So in this case, you kind of using that large amount of information across the genome to make kind of a selection in different populations. So it started with what we call as a training population. You create the population as your training. You characterize that population with sequencing. Okay, so you do the follow-up, you kind of creating characterize the fine tuning, and at the same time you also evaluate that for the traits that you are interested, and then from there you create the equation, and then this equation then applied to different population just by doing genotyping instead of phenotyping. So you have that genotyping data, you feed that data into this model that you develop. Okay, so that you make selection right at the very early generation in the breeding program. So it's been used in many places. So we are using this as well in our program. Okay, so in a way it looked like this. Okay, so the step how to do it, you set the training population, what we call training set, you genotype those and this population, and also phenotype those population, and you regress phenotype on the markers in the population, and then use that equation, regression equation, to predict the markers in novel germplasm, and then you can make selection, crossing, and then you have to refine this equation after a certain generation. Okay, so this is just an example how we do it with genome selection. So you have an elite germplasm pool here. Okay, you select the most diverse one as your training population. Okay, lines derive from a combination, some elite lines, some germplasm, and then you do phenotyping and genotyping, meaning that you characterize every single individuals in that population, and also you evaluate in the field or in the growth room or in the greenhouse, whatever condition, whatever traits you are interested in, and from there you develop a model. Yeah, and then you kind of going around back again, you can refine this model, okay, after certain years where you have more data. And then you can also have other population, untested population. So this population is not evaluated in the field, but you do the genotyping only. And then you use that model you develop to calculate what we call genomic estimated breeding value. So you make selection just based on this genotyping only. You make selection, you test the lines, and you release varieties. So this is really fascinating, and takes some bioinformatics, some resources, but this is how we cut the process of selection from 10 years to become like only 4 or 5 years. So cut in half of the time to release or develop a new variety. Okay, so initially this was published by the group from Australia, okay, from Mike Goddard group, and basically what they are doing is in animal breeding. Okay, so you can see that by using blob or Bayesian A, Bayesian B, it's a different kind of model in genomic selection. Okay, random forest, so kind of more like advanced statistic is needed. So he kind of allow him to make a selection as accurate as 0.8% accuracy. Okay. So we also just recently, in fact, this paper just released, just published last week. So in Nature, so I'm part of this group. We sequence 3,366 genomes of chickpeas, and then we develop a model. We kind of tell a story. So this, by the way, this paper is publicly available. It's open basis, so you can Google and then you can look into the paper here. Okay, so one of the component in this paper, oh, by the way, this is the picture of me and Rajiv Varshney, Rajiv from ICRISAT, India. He's the leading author in that paper and the leading kind of the coordinator for the project. So there are 57 people from 23 universities globally involved in that paper. So we developed in this paper, optimize the 3 genomic breeding approach, and we optimize it using haplotype-based breeding, genomic prediction, and selection contribution, okay, for developing tailor-made and climate-resilient chickpea varieties. So if you are interested, feel free to look into this paper. And besides that, there is other information that we published in this paper as well about the haplotype map, about how chickpea is shaping up from the center of origin to the other places globally. And also so-called we developed the pangenome in that chickpea project. So I'm really happy to have that paper now published in Nature with the impact factor 2020 is 49.9. So the other breeding techniques that already well known for many years now, what we call GM breeding or GMO. So in GMO breeding, basically you take the gene or gen in Indonesian, in Bahasa, from any species, even across a kingdom, and then you clone that gene, again, you clone that gene, and then you transfer into any species or most of the species that you want to try to improve. So this is kind of one way when there is no source of variability in the species that you are working with. So you need to create that variability by kind of taking from other species, from bacteria, from any organisms. Okay, so this is just simplifying that. So this is, and then the product of this is called as a GM. So you know, like a GMO product, okay, or from genetic transformation. So there are the the major rule of thumb, what we call, okay, what to use, the molecular breeding or use GM or both. Okay, so molecular breeding, basically you need to have population, you need to have genetic resources, and the plant should be sexually propagated, meaning you have to make the cross. With the GM, you don't need that cross needed. So okay, you can just use any source of gene and you transplant, and the plant can be propagated vegetatively in this case. Okay, so I like to focus the last part of my talk here about the gene editing. Okay, gene editing is kind of the new techniques that just recently, and then the scientists who developed the techniques, Jennifer Doudna and Jennifer Carpentier from France, they received Nobel Prize for this in chemistry for this technique. So basically, what is gene editing? Gene editing, basically, we make use the process that already happening within living organism. Okay, when the living organism within the cell, for example, exposed to stress, okay, there's a DNA break within the cell, and then the plants or the cell itself try to repair that break, okay, to fix the break happening. Okay, so this is kind of the essential, the basic technique. Okay, why, how do the gene editing? So because of that break, okay, the cell try to fix that break, and then kind of so that the cell is still functioning. Okay, so in this case, CRISPR-Cas9. Okay, CRISPR is stand for cluster regularly interspersed short palindromic repeats. Basically, this is from bacteria, how bacteria defend itself from virus, from a bacterial phage attack. So in this case, we design what we call a guide RNA. So guide RNA is kind of part of the RNA that will look into the genome that you want to edit, okay, find the homology. Okay, so and then the Cas9 is the enzyme that will cut that piece of DNA where you target to edit. It will create what we call a double strand break, okay? And then the cell itself will repair that break after you. So basically, here is that you directing, okay, which part of the genome, which part of the chromosome you want to fix or you want to edit. For example, you know one gene, you want to knock it off so that the gene is not functioning. So you create a stop codon, so you know exactly where you want to create that editing. So this has been used widely. Okay, so this guide RNA is only short, about 20 base pair, and then you kind of mix that with the Cas9. And then this guide RNA, repeat again, will find the homology within the cell, in the chromosome, and then the Cas9 will cut it, and then the cell will fix it. Okay, so during the fixing process, this is when kind of you modifying it. Okay, you just leave it up to the cell to fix it. Basically, they will create a random insertion or deletion that we call it, could be like a gene knockout. Or during this process, you can also give what we call homology direct repair. Basically, you insert a template so that the cell, when they kind of try to repair that to create a new strand, will follow this kind of template. So this will be precise editing, but low efficiency. Good. So a lot of papers now available about talking about this gene editing. So it can be used in many ways. Okay, in plant breeding, site-specific DNA integration, pest resistant, enhanced nutritious plant, disease resistant, drought resistant, viral resistant, tasty vegetables, changing the flavor, and genome engineering. Okay, so this is very versatile techniques. Okay, in the process, you can kind of basically, this is the main step in the gene editing. You need to identify the target gene identification and design the experiments. You need to do plant transformation, how you kind of transform that part into the cell. You do the molecular analysis, make sure that you have the mutation or the editing that in the target genome that you're looking for, and then you grow that cell into intact plants. Okay, so this molecular analysis to make sure that the mutation or the edited part of the genome is exactly what you want it to be. Okay, so this has a broad applicability across crops, across species, across different traits, across the genome size. So all the way from Pinus, from pines, populus, small, and humans in sugarcane, in barley, okay, avena, so oat and triticums and oryzes, typha, rice. It's all been done. Okay, so and then this also allows multiplexing. So okay, you can edit more than one genes at the same time. For example, in maize, they targeting like 3 genes here, legal as a gene region, male fertility gene and male fertility gene on different chromosomes at the same time. So it was published in 2015. And there's another example where a group from Purdue universities basically editing about 12 to 15 genes at the same time. So the gene related to abscisic acid receptor, which is related to plant response to stress. So they modified what we call pyrobacterium resistant like PYL1. You can see here, and then the rice product from this gene editing increase the yield and can withstand more kind of adverse condition, still maintaining the yield and increase by 33%. So it was really fascinating with this technique. It was published in Proceeding National Academic in 2018. Okay, and this is another one. Okay, so this is basically a clonal of rice. So we have developed hybrid rice, but with hybrid rice, you need to produce the F1 every single generation every time. So this is making the rice, the hybrid rice, become like a vegetative. Okay, so basically you make the rice so they control during the meiosis. So they call the mime genes. Okay, they edited so that the F1 kind of maintaining. So you don't have to kind of making the cross all the time with this clonal hybrid rice. Okay, so this is just another example related to that. The other one is that editing the wild tomatoes from Solanum pimpinellifolium in these 5 genes, okay, the plant architecture, the length insensitivity, and large fruit size, and increased vitamin C levels. Okay, instead of in nature, it takes many years to kind of domesticate this trait into what we call as a modern tomato. So by gene editing, you can edit all those domesticated genes all together at the same time. So you can modify it as a proof of concept here. They kind of accelerate in one step from the wild into the cultivated tomato through the gene editing. Okay, and also this is what we call low glycemic index rice. Okay, so glycemic index, that for the diabetic people is they like to have a low glycemic index. So by modifying the starch branching enzyme SB genes, we can modify high amylose content and resistant starch in rice, so becoming more suitable for those with diabetics. And also brown plant hopper, so editing for stress tolerant, and in this case for resistant to insects, also been done. Okay, basically they modified the cytochrome P450 that encodes kind of in the susceptible line, so making the plants kind of reversing it from susceptible to become tolerant into this brown plant hopper rice. Okay. And also a number of other papers are talking about the disease in rice and in other crop using this gene editing. So and then by weeks, this paper related to CRISPR gene editing is coming kind of a lot of. So we also reviewed the technique in legume crops. So I have this paper, so part of the co-authors, if you are interested. Again, this paper is also open access. You can Google and you can look at the OI here number. And if you're interested, you can read it, and you have any question, you can let me know. Okay, so with all this technology, okay, marker selection, gene identification, and genome selections, and now gene editing, I'm sure that this will offer breeders the unprecedented ability to quickly develop important plant varieties that we can withstand and stabilize, increase the yield despite of the changing climate, can withstand pests and disease with fewer input, and maximize resource use efficiency, water, nutrient, etc. So with this technique, I'm kind of optimistic that we can increase global food production and food security. Okay, so with that, this is part of my group. Okay, a number of students already graduated back to their countries or working somewhere, PhD students, and then a number of postdocs and technicians working with the chickpea and flax group. And the funding is coming from the Grower Association, from the government of Saskatchewan, from university, and Western Green. Thank you very much for your attention and for listening to my talk. And I'll be happy to answer any question if you have. Just note that if you kind of hesitate, you can also speak Indonesian, speak Bahasa, so I'll be happy to answer that as well. Thank you. I see a question here on the chatting. Could you please share information regarding identified food crop genome tolerance to abiotic stress, especially drought? So when people are talking about the abiotic stress related to genome editing or other part of the crop, so they've been working in many different aspects of the drought itself. Okay, so some of the component they do looking at into morphology, looking at the root biomass, and some others looking at into the osmotic. So yeah, there are a number of papers that you can see that in corn, for example, they're already using that a lot, and even the company, there used to be like Pioneer Hybrid, and now under BASF. Okay, so they've been working. So there's a lot of things that already done in the drought in corn, soybean. Even like a simple trait in a breeding program, for example, stay green genes, okay, where the plant kind of maintaining less wilting under high water potential or under dry conditions. So that's also been done. So yeah, there I can give you can email me, okay, and I can share it to you directly if you like, Pak Kuku, ya. Terima kasih. Any other?
To ask the question. Ya, Pak Fatah. Please, Pak Fatah, you can ask the question here. Pak Fatah? Di chat, Prof. Kukuh and Pak Agus Mulyana from IPB is already answered by Prof. Taran.
Okay, Pak Fatah, I think I see you raised your hand. Please, if you like to talk, please introduce yourself. And if you want to speak Bahasa, fine too. Or even if you want to speak Jawa,
Pak Fatah, it's okay to speak Indonesia. Ya Pak, silahkan bahasa Indonesia, it's okay. Pak Fatah? The chat is already answered by Prof. Taran. Yeah, Prof. Kukuh and yeah. Okay, maybe another participant. Okay. Prof. Kukuh, please. Masih mute. Host, please. Pak Kukuh. Please, Prof.
by the water level. Okay, so when it's switching off, switching on by the water level, it just goes. It's not extensive but longer. So for us in agriculture, Prof. Kuku, the main thing is that we are not biologists. Biologists looking at it to the survival model. In our agriculture, we look into more economic. What is the threshold that we want to reach? Okay, that's different approach. Okay, for us. So at the end of the day, in my program, we look at more into the stability. How much a variety can withstand under certain level of moisture, still giving you the threshold of the yield that you still be happy to grow that. Okay, that's that. So then we look at into that, and then when we find that condition, and then we try to look at molecular physiology, there are a lot. It's not just single or one or two, three. Hormone levels, yes. Abscisic acid level, yes. Osmotic. Kind of, you need to know what is the yield potential, and then you need to expose that under stress, and then which one that allow you to make that selection.
Okay, thank you, Benjamin. Because previously I'm in plant breeding and plant genetics,
Okay.
WW Medicine, work with William Tracy, Dr. William Tracy.
Oh, Bill Tracy in Wisconsin.
Yeah.
Oh,
you guys.
yeah. I was in Wisconsin in 2000, Prof.
Oh, okay.
Yeah, in 2000, but I was in the not in agronomy. Bill Tracy is in agronomy, right?
Yeah, you're right.
Yeah. I'm in the genetics building just across.
Oh, yeah, I see.
Henry Mall, from Henry Mall.
Okay.
Yeah.
Okay, yeah. Can you just remind me around 30 years ago.
So you were there before me. I was in 2000. So
Yeah,
20,
you're right.
21 years ago. You 30. So you must be all around the Eagle Heights.
Yeah, you're right. Yeah. Okay, thank you, Benjamin.
Thank you.
I'm waiting for your paper regarding the abiotic stress. Could you send me some paper? Yeah.
Will do.
I write down my email address here. I don't know.
Okay, maybe you can give it to the committee, to Ibu Fadila, and then she can or Ibu Fadila can forward my email to.
Okay, thank you very much, Benjamin.
You're very welcome. Well, if there is no more question, I think that's what I can share again, and I'm happy. If you have any question, you can email me. The committee has my email, and I'll take a note. Thank you very much for your attention.
Now we can step to the gift certificate for Professor Taran from our directors, Dr. Sarono. Because at Canada now it's still midnight, isn't it?
8:45 to 9 p.m. So.
Oh, thank you very much for your concern to help these keynote speakers. I think it's really, Pak Sarono.
Okay, thank you. Thank you, Prof, for your willingness to be a keynote speaker at ICOS 2021 at Polinela.
My pleasure, Pak. Senang bisa berkontribusi.
Oh, iya. Thank you very much. Jika punya waktu, kapan-kapan bisa ke Lampung, Prof.
Dengan senang hati, Pak. Pasti akan saya kunjungi.
Okay, thank you very much.
You're very welcome.
Being as a keynote speakers in ICOAS 2021 at Polytechnic Negeri Lampung. Hopefully in the future we could bring together to the great opportunity in international conference. Thank you very much. Big applause for Professor Bunyamin Tar'an. And then we can go to go step to the parallel sessions with the two keynote speakers from Penang, Malaysia, and from Polytechnic Negeri Lampung. Firstly, I would like to introduce the speakers, Professor Rosma Ahmad. Professor Rosma Ahmad, she was at Bioprocess Technology Division School of Industrial Technology, University Science of Malaysia. Professor Rosma, are you already here?
I'm in. Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. The sounds of your voice is so.
I can hear you very clearly.
Okay, yes. Thank you for join this prominent sessions, Prof. Rosma. Allow me to inform your curriculum vitae. Professor Rosma Ahmad, she was a graduate bachelor of food technology bioscience from University Science Malaysia, Penang University at 1993, and then finished the Doctor of Philosophy from Bioscience and Biotechnology, Strathclyde, Glasgow, UK. At her experience, firstly as the Dean, Deputy Dean, especially in academic affairs, and also the program chairperson for food technology divisions. Her publications, she's so productive in past 9 years, produced 20 reputable international journals, 6 book chapter, and also 7 paper at conference proceedings. She also holds as project leaders for 8 grants and 18 as co-researchers. In this occasion, she will bring the new opportunity for economic potential and application for Dioscorea hispidae. This is our introduction about Professor Rosma Ahmad, and then let's hear together the presentation from Professor Rosma. Please, time is yours.
Okay, thank you, Bu Fitri, for the introduction. Actually, I was invited for this conference by Ibu Marlinda. We met at 2nd ICOFA 2019 in Bali. The field of research is actually in food bioprocess technology. I think we had a misunderstanding with the secretariat because the title given to me was only last week when I already submitted the abstract for the title. So, but I think it's still relevant to this conference, yeah, about this Dioscorea hispida. It's a tuber. I also prepared a slideshow with an audio because I think internet line may have a problem. So I wish Secretariat could share the slides with audio with the audience.
A co-host, so you can share the video.
So the Secretariat, do you have problem sharing the video? The slideshow, actually not the video, the slideshow, PowerPoint slideshow.
You already co-host, Prof. Rosma. You just
I already
when you
send the slide to the Secretariat. So do you want me to share from my side?
Okay,
The
okay.
Secretariat?
Wait a moment.
Secretariat?
Wait a moment. Yes.
There is lagging here. The internet is lagging in my side.
Oh yeah, yeah. I see. Talking about the Hispidie at Malaysia, these commodities is already cultivated by farmer?
No.
No, not
it
yet.
is widely
widely
available.
plants.
You cannot purchase in the market.
Oh yeah, I see. Usually this is the wild plants in the backyard.
Yeah, correct. But there is effort by the country to cultivate this tuber.
Is
because
there
of
any
the
cultivar?
potential. There are plant breeders looking for the breed, the breed of this cultivar.
Okay. Oh, I'm sorry. We didn't get the video that you.
Okay, so I
please
share
send
from my
to.
side. I hope the line is okay.
I hope so.
Okay.
The slides look like a good presence. So you can continue the presentations.
Okay, thank you very much. Honorable Director of Polytechnic Negeri Lampung, Indonesia, distinguished guests, distinguished fellow keynote speakers, Yang Berusaha Ms. Fadila Margasati, Chairman of the Organizing Committee and her hardworking committee members, fellow participants of the Second International Conference on Agriculture and Applied Sciences, ladies and gentlemen, Assalamualaikum and good morning to all. University Science Malaysia, I am honored to be invited to this conference. However, it is sad that we cannot meet and I miss to view the beautiful scenery of Negeri Lampung. Today, I'll be talking about Dioscorea hispida, its economic potential and applications. I've been researching about D. hispida starch since 2013. And although this tuber has produced significant amount of starch, it has no economic value. So hopefully after my talk today, everyone here could appreciate this unknown or forgotten tuber. Okay. Before I talk about D. hispida, let me introduce University Science Malaysia, my working place for the last 22 years. USM was established in 1969, the second oldest university after University Malaya. USM has 4 campuses. Main campus is in is in Pulau Pinang, a state in Malaysia. The Penang Island, also famously known as Pearl of the Orient. Another campus is for the engineering programs, is at the mainland near border of Perak State. The third campus is for health, medicine and dentistry programs in Kelantan State of Malaysia. The fourth campus caters for the advanced research in medicine and dentistry, located at northern part of Pulau Pinang. USM offers 71 bachelor's degree programs and 278 postgraduate programs by coursework mode, mixed mode or research mode at master and PhD levels. There are 22,878 undergraduate students and 11,251 postgraduate students enrolled in various programs in 2020. USM has 1,886 academic staff from 26 schools. Okay. Dioscorea hispida, commonly known as yam, belongs to Dioscoreaceae family. It's a perennial monocotyledonous tuber producing climbing plant, as indicated in the figure 1, indicated by arrow in figure 1. Okay, this plant naturally thrives in tropical and subtropical regions of Philippines, China, Taiwan, New Guinea, Malaysia, Fujian, Bhutan, Indonesia, Bangladesh, Sikkim, Thailand, Cambodia, Laos, Myanmar, Vietnam, and Africa. There are 1,137 species of Dioscorea, but only about 600 species are edible, and D. hispida is one of them. Each mature plant of D. hispida can produce up to 15 kilograms of starchy tuber. Okay, the tubers is shown in figure 2, which however contains water-soluble neurotoxic alkaloid Dioscorin. The presence of Dioscorin has negatively impacted the value of the starchy tubers. Fortunately, traditional method of steeping the tubers in flowing water can successfully remove the water-soluble toxin to a safe consumption level. And 1 kilogram of fresh tuber can produce up to 270 grams of starch powder. Therefore, the economic value and potential applications of the hispida tuber starch will be explained in this talk. Generally, there are 8 steps involved in the production of the hispida starch. The starch is processed from the hispida tubers following these steps: washing and peeling the tubers, slicing the tubers into thin slices, steeping the slices in 5% of sodium chloride solution for 3 days, grinding the slices, separating the slurry, collecting the filtrate, sedimentation of the starch, and drying of the starch. The proximate analysis in dry basis of the hispida starch in comparison with other commercial starches, sago, potato, and corn is shown in table 1. The hispida powder contains 0.42% total crude protein, 0.23% total crude fat, 0.47% ash, and 98.88% carbohydrate. The carbohydrate was mainly starch, 88.8%, with amylose to amylopectin ratio of 28:72. Ash content of the hispida starch was similar with potato, but higher than sago and corn starches. Ash content is an indication of mineral content of a food. This therefore suggests that the hispida starch could be an important source of mineral. The granules of the hispida starch were observed at 5,000 times magnification using a scanning electron microscope, as shown in figure 3. The granules were polyhedral shaped with coarse surface without fissure. Granules of sago starch is shown in figure 4, and other starches, corn, potato, tapioca, rice, and wheat, have oval shape with smooth surface. So the granule structure of Dihispida tuber starch is very unique. Okay. The average diameter of Dihispida starch granule was 2 to 4 micrometer, the smallest granule if compared to other species of Dioscorea, 23 to 40 micrometer. Dihispida starch granules was also the smallest as compared to that of sago, 20 to 40 micrometer, potato, 15 to 75 micrometer, and corn, 8 to 12 micrometer. The compact and small granules make Dihispida starch has higher gelatinization temperature than the other starches. There are some potential applications of Dihispida starch in non-food industries. Some, but not limited examples, are discussed here. Okay. Material in thermoplastic film. This has been researched by a group of researchers in University Putra Malaysia, UPM. Okay. Chemical composition and physical property of starch and fiber waste from Dihispida tuber were analyzed for the suitability as filler material for biodegradable thermoplastic film composite. Okay. The experiment revealed that the starch and fiber have excellent thermal stability and high decomposition temperature of the 309.7 degrees Celsius and 315.4 degrees Celsius, and crystallinity index of 27.5% and 39% respectively. It was concluded that starch and fiber waste of Dihispida tuber could be promising alternative sustainable biomass materials as renewable filler materials in food packaging application. Okay. The other application in non-food industry as coagulant in wastewater industry. This has been researched by a group of researchers in engineering campus USM. Okay. Starch from the Dihispida tuber is used as natural coagulant, an alternative to environmental detrimental chemical coagulant in textile wastewater treatment. Okay. Textile mill wastewater is a high turbidity effluent which contains large content of dyes, significant amount of suspended solids, and contributes to high COD concentration. Incorporation of 2.5 gram Dihispida starch per liter of textile mill effluent at pH 7 managed to reduce or remove COD, turbidity, and color of the effluent by 28%, 94%, and 64% respectively. The other non-food application is as an antifungal agent, researched by a group of researchers from Universiti Kebangsaan Malaysia. Okay. In wood decaying prevention. Okay. An antifungal coating film containing Dihispida tuber starch to prevent wood decay was developed. Rubber wood coated with a modified film coating formula with a ratio of 2:1:1 and 1 polyvinyl alcohol, starch, sorbitol, hexamethylene tetramine was able to retain approximately 50% of its original weight. Moreover, the coating film was effective to inhibit the growth of 2 wood-rotting fungi. The other application of this food, Dihispida tuber starch, is as component in water filtration membrane. This has been researched by a group of researchers from Syiah Kuala University, Banda Aceh, Indonesia. Okay, Dihispida starch was used as the material in water filtration membrane. This was achieved by constructing a forward reverse membrane made of a glutaraldehyde cross-linked ketosan and the dehispida starch into a fabricated water filtration bag. Okay, it was concluded that the filtered brackish water using the filtration bag with ketosan dehispida starch membrane had met the quality standards for drinking water by WHO. Dehispida starch has been researched to be used as food ingredient in replacing a certain percentage of wheat flour in the making of muffin, cookies, and bread. And these food products have received good customers' acceptance. Therefore, dehispida starch has the potential use as a food ingredient in bread and confectionery. Okay. The other food application of dehispida starch as a glucose syrup. Dehispida tuber starch can be further processed to glucose syrup. The glucose syrup was obtained by treating the gelatinized dehispida starch with a consecutive action of alpha amylase and glucoamylase. Okay, starch polysaccharides consist of amylose and amylopectin. Amylose is a linear polymer of glucose linked by alpha-1,4 glycosidic bonds, and amylopectin is a branch polymer of glucose linked by alpha-1,4 and alpha-1,6 bonds. Okay, alpha amylase hydrolyze amylose polymer into oligosaccharide and maltose. Reacting the starch polysaccharide with alpha amylase for 60 minutes had increased the total reducing sugars concentration as maltose by 148%. The mixture was consecutively reacted with glucoamylase for 40 minutes, and the final mixture had a total reducing sugar concentration of 4.42 milligram per milliliter. Glucoamylase hydrolyzed disaccharide maltose to glucose monomers. As a result of the enzyme's reaction, the starch polysaccharide were fully hydrolyzed to glucose and fructose, as determined by using a HPLC system. Dehispida glucose syrup had a dextrose equivalent value of 42%, with 99.7% of the sugars were glucose and 0.3% was fructose. Another application of dehispida starch is as oligosaccharides. Dehispida oligosaccharides was obtained from the starch hydrolysis with sequential action of alpha amylase and pullulanase starch for 6 hours. The yield of oligosaccharide produced was 19.3%, 19.3 milligram per gram of starch, and mixture with degree of polymerization of 227 was obtained. The oligosaccharides were evaluated for prebiotic potential. And prebiotic material is a non-digestible carbohydrate by gastrointestinal tract digestive system, but fermentable carbohydrate by beneficial intestinal microflora. The oligosaccharides were subjected to gastric juice digestion at pH 1 to pH 4 for 180 hours and to gastrointestinal tract digestive enzymes, salivary alpha amylase and pancreatic pepsin and pancreatin. The oligosaccharides were partially digested by gastric juice at pH 1 and pH 2, but resisted the digestion by the gastric juice at pH 3 and pH 4. At least 64% of the oligosaccharides were still intact after subjected to digestive enzymes, and more than 50% of the dehispida oligosaccharides retained in the dialysis tubing, indication of its resistance towards acid digestion, digestive enzyme hydrolysis, and intestinal absorption. Incorporation of dehispida oligosaccharides in the culture medium of selected beneficial bacteria was able to enhance the bacterial growth, reduce the pH medium, produced substantial amount of lactic acid and propionic acid and exhibited positive prebiotic activity score. In conclusion, findings from the oligosaccharides of the Hispida tuber starch have the potential to be used as a dietary prebiotic ingredient. Although the pharmaceutical benefits of D. hispida tuber have not been scientifically proven, native people around the world has been using the tuber for eye treatment by applying the eye with water or soap tuber, food skin treatment, by pasting the tuber on the affected parts overnight to treat the peeling of skin, treatment of vomiting, indigestion, and used as a purgative, wound and injury treatment by applying roasted tuber paste on the affected area. Kumar and co-researchers had studied on the ethnopharmacological potential and traditional use of Dioscora species by the local people of Similipal Biosphere Reserve, Odisha State. Species tuber known as tape in Indonesia, tapai in Malaysia, with red mole, usually is Monascus species, has been proven to have anti-hypertensive, anti-diabetic, anti-lipidemic, and anti-cancer activities. Fermented Dioscora tuber as anti-hypertensive agent. Okay, research by this Wu et al. Yeah, in 29. Okay, systolic blood pressure and diastolic blood pressure of hypertensive rats orally administered with 75 milligram fermented Dioscora per kilogram of body weight of the rats for 8 weeks decreased significantly. Administering 150 milligram per kilogram of fermented Dioscora to the rats had significantly reduced the blood pressures after 8 hours of administration. This is because the fermented Dioscora contains acid, anti-inflammatory yellow pigments, monacin and acaflavin, and angiotensin-1 converting enzyme inhibitory activity. Okay. Fermented Dioscora tuber as anti-diabetic and anti-stress agents. Fermentation of Dioscora tuber with red mole produce yellow pigment monacin. This pigment show anti-diabetic activity in streptozotocin-induced diabetic rats. Blood pressure, blood glucose, and serum insulin of the rat, which were fed with 30 milligram of purified monacin per kilogram of rat's body weight per day for 8 weeks, was lowered and increased respectively. The same group of researchers also studied the effect of monacin from the fermented Dioscora on the lifespan of C. elegans, a worm under high glucose condition. Monacin increased stress resistance and expression of the stress response genes, thus extended the lifespan of the worms. Okay. Another beneficial pharmaceutical benefits of fermented D. hispida as an anti-lipidemic agent. Okay. Monacolin K, lovastatin, is a secondary metabolite produced by Monascus perspirus during the fermentation of D. hispida tuber with red mole. Monacolin K is an inhibitor for HMG-CoA reductase in cholesterol biosynthesis. Fermented Dioscora exhibited higher anti-cholesterol activity than fermented rice because monacolin K production was higher in the fermented Dioscora. Yellow pigments, monacin and encapflavin were produced during the fermentation, and also shown significant effect on lowering cholesterol, triglyceride, and low density lipoprotein cholesterol levels in the blood serum and alpha lipid plaque of the tested hyperlipidemic rats. Last but not least, the pharmacological benefit of fermented Dioscora tuber is as anti-cancer agent. Okay, ethanolic extract of fermented Dioscora tuber with red mold was tested for its anti-cancer effect on cancer-induced hamster. The carcinogenesis of the buccal pouch of the hamster was induced by DMBA. The ethanolic extract had shown therapeutic potentials against oral cancer, as it had decreased nitrite oxide reactive oxygen species and prostaglandin E2 overexpression, and simultaneously increased P3 serum tumor necrosis factor alpha and interleukin 1 beta to significantly stimulate caspase 8 and 3 activities, indicating that the extract had managed to reduce oxidative damage caused by the MBA and induce apoptosis in the oral cancer cells. For the conclusions, there are 3 conclusions can be derived from this talk. Number 1, the Hispida tuber has gained great attention due to its starch content. And number 2, starch of the Hispida tuber has potential use in food industries as starch, food ingredient, as oligosaccharide, and as glucose syrup, and in non-food industries as material in the making of thermoplastic film, as coagulant, as antifungal agent, and as component in the water filtration membrane. And last but not least, the final conclusion is secondary metabolites from fermented D. Hispida tuber with red mold have been researched for their pharmaceutical benefits as anti-hypertensive, anti-diabetic, anti-lipidemic, and anti-cancer agents. So in all, what we can say that the Hispida tuber has great potential in industries. So the application of the Hispida tuber is also varied. Okay, these are the references for the papers, and thank you for your attention. Bu Fitri, I pass back the session to you, Bu Fitri.
Can you hear me?
Yeah, I can hear you, Bu.
Yeah. Thank you for your great presentations. Then we go to the questions session. It's okay, Prof. Rosma?
Yeah, I'm okay.
Okay, please, all participants, if there's any question for Professor Rosma, please, you could raise hand button. Press the raise hand button if you want to ask the question directly to Professor Rosma. Or type the question in chat box. Probably you could explain more about on your bioprocess technology, use the Hispida. Have you any experience about that?
My research food bioprocess technology. So the food application examples are actually from my research. Okay, we use enzymes to prepare the product that we want. Okay, we increase the yield of starch extraction from the traditional method of starch extraction using enzymes. And we also use enzymes to degrade the starch to produce oligosaccharides as prebiotic material. And we also use enzymes to degrade the starch to produce the glucose syrup. So that's where bioprocess technology is in the picture. We use enzymes and fermentation. Currently, we are researching on the fermentation of the tuber for the pharmaceutical benefits as well, but using a Malaysian ragi, Malaysian mold.
Okay, there's one question in chat rooms.
Okay, actually,
From
because the toxin is water soluble, so it is easily removed by soaking the tuber slices in water. And traditionally, people soak these tubers in the river, free-flowing river. But in industry, we use, a group of researchers in another university in Terengganu. They developed a machine with a spiral water movement. So they can reduce this toxin. And when we check the alkaloid level of Dioscorin in our center, poison center, it is below detection limit of 1 ppm. So it is it can be easily removed. Yeah, the toxin can be easily removed by soaking in water.
Okay. The question from Ibu Sintawati, lecturer of Polytechnic Negeri Lampung, about the treatment to remove the toxic material in the already answered by Prof. Yosma. And the one's question from the student of Polytechnic Negeri Lampung. I can see clearly. Sorry, it's my. What about the protein? I can see the. I'm sorry. To reduce protein content or not, the process of the remove the toxic material, is it influence the protein contents or not?
This is a question by Ike, student of Polytechnic Negeri Lampung.
Ike.
So yeah, we also call it ubi gadung, same name, gadung.
Oh, same name, ubi gadung.
Common
Yeah,
name,
Indonesian
gadung.
calls ubi gadung.
Correct. To be cheap was soaked in water until 3 days. Yeah, correct. So you want to know that process. Does the process reduce the protein content or not? The main thing that we want from this gadung is actually the carbohydrate, not the protein. Okay, but as you can see from the slide just now, the protein has 0.42%. Was it?
Yes, right. 0.42%.
Yeah, 0.42%. So it's very little amount. So it's not a good source of protein. It is for starch. Okay.
Okay.
All right.
Okay, I think the discussion between the all participants with Professor Rosma in this session, it's so interesting. And the next step is the our grateful with gift certificate from Director Polytechnic Negeri Lampung. Dr. Sarono? Or Miss Fadila Margasati as oh Pak Sarono is already here. Please, Pak Sarono. Pak Sarono unmute, Pak.
Oke, Bu. Oke, thank you to Professor Rosma. Hello?
Thank you, Pak. Thank you,
Uh,
Pak Sarono.
Oh yeah, to be a keynote speaker at EQUEST 2021 at Polinela, I will give you a certificate of graduated.
Thank you, Pak. Thank you.
Okay, thank you very much.
As the keynote speakers in International Conference of Agriculture and Applied Science 2020. Hopefully in the next years or maybe in another our grateful for your presence here. Hopefully we could keep in touch for the next scientific conference. Thank you so much. And then we go to the next parallel sessions. Professor Ryoi Kada from Yonawate Gakuen University, Japan. Professor Kada. Halo, Profesor Kada.
Do you hear me?
Yes, clearly. Thank you so much for joining the International Conference of Agricultural and Applied Science in Polytechnic Negeri Lampung.
Thank you.
We're so grateful for
Yes.
your having you between us here today for talk about the environmental
Yes,
risk management.
I'm very happy too.
Yeah, Professor
I'm very happy
Kada
to
has
join.
long story
Yes.
in collaboration research with the University of Lampung, especially with Professor Bustanul Arifin and also Dr. Hanung Ismono. It's
Thank you. Yes.
our honor having you
Professor-
here.
Yeah, Professor Ismono and Professor for many years, and I visited many places in Lampung and other places in Jakarta and in Indonesia. I enjoyed it so much.
Hopefully in the next
Do you hear
occasion,
me?
you could attend to Polytechnic Negeri Lampung.
Oh yes,
Yeah.
of course. I really long for it and really expecting
Okay,
to join. Thank you to see you. Thank you.
Professor Kada, you would like to
Yes.
start the presentation?
Well, I don't know. Have you received my PowerPoint paper
No, not
of my
yet.
presentation?
You could
I hope
share.
you received it.
You already sent it?
Don't know how to do it. How can I do
You
it?
could share,
This moment.
choose the button of share screen and choose your paper
Yeah,
file.
send screen.
Yes.
Send screen.
Oh yes,
Ooh.
we already got the papers. So the presentator's slide will
Papers.
be shared from here.
Oh, okay. Can you post? I mean, can you show it?
Yes, yes. One moment.
In this program, I don't know
Yes.
how to do it.
One moment. For information, Professor Kada is also our my examiner in dissertations.
That's right. That's right. Many
Thank you
years
so
ago.
much for your support
Congratulations.
to finish my doctoral study.
That's right. Yes. Well, Kyoto. I live in Kyoto,
why you
Japan, which is winter now.
Oh yeah, start the
Very
winters.
cold.
Hopefully you always healthy and happy.
Yeah, I hope to visit Lampung, Indonesia, so that I can enjoy.
Yeah, we miss you much.
Hot weather and climate. Well, anyhow, how can I show my paper? I don't know.
No,
Could you
we
please
do.
show?
We do from here. Just wait a moment.
Yes. Okay, please.
Yeah. Still on process.
Okay.
We have wonderful
Can you show it?
invite speaker in this occasion, Prof. We got Professor Bunyamin Taran from Canada, Professor Rosma, and also you, Professor Kada Sensei, my sensei, and also Dr. Anung
Yes.
Wahyudi from Polytechnic
Yeah.
Lampung. It's okay with the
Oh,
presentation?
okay. Yeah, if you can show some of the pictures, I've sent it to you. I don't know how to post it in this picture.
Actually, you are already a co-host, but I think the committee still prepare. Please,
Preparing now?
Yes.
Okay.
Yes, this is starts now.
Mm-hmm. Yes, please.
Yes. The slideshow starts.
Okay. May I start my? Yeah. May I start
Yes.
my presentation?
Please, the time yours.
Okay. How to? Yes. Could you come back to the slides? Yes. Can you show it? I cannot see my slides. Do you see it? Sorry.
Yeah, still a little bit problems. Oh, because
Yes,
the
that's
file
right.
is so heavy. Because many picture.
Maybe.
Yeah.
Yeah, so many
You
pictures.
have many
and,
fascinating
yeah,
photo
color.
documents for the how to take good care the environmental risk. So I think this
Yes,
is
that's
make
right.
a little bit problem to show the file.
Yeah, I hope.
Would you like
hope
to try
You can show
by
some
share-
of my
yeah, from your side.
slides. Yes, this is the slide, the first page on my slide. Can sorry.
Still, please be
It's not
patient
working.
with the inconvenient situation.
Very sorry. Oh yes.
Yes. Yes.
Can you hear me?
Yes, clearly.
Okay.
Please.
Okay, it's clear now. I'm very happy to have this opportunity. And do you hear me, by the way? Do you
Yes.
hear me?
Yes, yes, clearly.
I'm talking. Okay, I am talking. I'm talking from Kyoto, Japan. Do you hear me?
Yes, Prof.
Okay, okay. May I start my presentation?
Please, you could start.
Okay, yes. And the title is Green Infrastructure and Sustainable Resource Management. I mean, green resources are very important, but the situation now is in many places, it is not well maintained or in dangerous conditions. Okay, let me move to the next slide. Could you put it to the next slide? Change to the next slide. I don't know how to do it.
It takes time.
Oh, okay.
Yes. It's already the next
Yes.
slide.
The next slide. Oh, this is a front page. Yes, okay. The point is, in many Asian agricultural scenes, ecological risks are expanding, and we need to focus on various ecosystem services. We classify into 4 types of ecosystem services. And next, please. So we have lost so many agricultural land, and that will affect how we can catch fish and other agricultural production. Food system and how to eat will be also changed by such natural resource, conditions. Okay, this is a case of Luzon Island in the Philippines. People are eating from natural resources. Thank you. Next, please. So what we did is we took water sample and fish samples and sediment and water, so that we can see how natural resources will be affecting what we eat and human health conditions. Okay, those research relations have been analyzed. Okay, thank you. Next, please. Next slide, please. Okay, this is how we collected samples from Lake Laguna, Laguna de Bay, in southern Luzon Island, the Philippines, which is very, very important food resources for the people nearby in Manila too. Okay, next please. We took water samples and plant samples and also insects and how, sediment, water, and natural resources are related. Plant, insects, they are all related. So we took all the samples from various places. Okay? And we examined, how they are polluted or are endangered, those kind of things. Relationship. Please, next, please. And we analyzed at the laboratories with many equipments, chromatography and other research equipments, and by natural scientists and also human scientists as well. Water quality analysis is very important and have been conducted at the Research Institute at Kyoto. Thank you. Next, please. Okay, in the case of Laguna de Bay in the Philippines, we took many samples from all over Bay areas, from Manila to the southern rural areas. You can see from this map, the samples of river water, is how they are polluted, by the degree of human settlements and industrial activities. The more, industrial activities and human, pressure is great, water is polluted, and that's quite natural. And so we need to maintain rural and agricultural resources, as much as possible. That's one of our conclusions. Please, next, please. We took the cadmium concentration, and in many places there are some, because garbages and garbages are one of the main sources of pollutions. So we need to be very careful, even in rural areas, how to deal with such, garbage collection and garbage disposal. It's another point of keeping water and natural resources clean. Okay, thank you. Next, please. Okay, this shows the pathways of pollutants from upstream to downstream. Now, ecological risks are here and there, and from time to time, lakes are polluted, and very heavy pollution sometimes kills fish, so much of fish, and people are damaged. I mean, eating fish have been prohibited or even, damaged because of this, water pollution. And that has happened in many years back in Japan too. So we need to how to clean the water, how to maintain water, cleaner as much as possible. Okay, thank you. Next, please. And with University of the Philippines at Los Baños, UP Los Baños people, we have been working together, working with fisher folks at Laguna Lake, so that people can be helping to take the samples. And sometimes we held workshops, meetings with the community people, so that we can obtain to understand local knowledge as the key elements of improving the conditions. Thank you. Next, please. Sometimes we interviewed with the local people how water is running and how wind is affecting the water quality from point to another point. Because, sometimes fish kill has been a very serious issue in the lake, so that people are so much damaged by such fish kill, by pollution of the water. So we need to trace where such pollution comes from, by the real sources of such pollutions, so that we can control how to do it for the next generation. Okay, thank you. Next, please. So biosignal mapping is very, very important. You can see in this slide that we have shown different biosignals: water quality, smell, water table, conditions of natural resources, fish, shrimps, and presence of chemicals, and those kind of all the data by different points, and we pinpointing how such data will be shown in the map. Mapping of such very powerful to try to show the people, the local people, how pollution comes from and how pollution can be improved, by human actions. Okay, so we have, picked up such biosignal data and shown on the map. Thank you. Next, please. Okay, On the map, we have shown, on different data of fish kill, and the conditions of plants, and how human activities are affecting on those natural resource conditions on the map. It's very powerful and to persuade people, how human activities, industrial activities are affecting such natural resource conditions. Okay, this is one of the case studies at Laguna de Bay in Luzon Island. Ecological risk mapping is one of the very powerful scheme to show the natural resource conditions. Okay, next please. So we have conducted the risk assessment of oxygen, water, and sediment, bottom sediment, and they are related. And so we picked up, such samples for and the degree of oxygen, density, and sediment quality. They are related quite much. So the long-term human activities will be affecting the sediment of the area and water and biological resources like fish and water plants as well. Okay, so they are all related. Next, please. We asked the local people to help for data collection. We asked the fishermen to help what kind of fish, how much of fish can be caught in relation to, and how, water, temperature changes in the day, during the year and months. Those are related to the conditions of fish catch as well. And so the collaboration with the fishermen is very, very important to collect the data and analysis as well. Thank you. Please. Next, please. Okay, these will show large-scale mapping of fish resources in the lake, and how each locality has, fish resources in relation to water quality. Please, fish kill. Because, fish kill has been taking place so often in the lake because of human settlements. And also, in contrast, local people have some traditional wisdom and knowledge how to, keep fish resources and water as clean as possible. Okay, this is one of the adoption of traditional knowledge. They call it Yang Cow system for rehabilitation of fish resources and fish catch. And this type of traditional wisdom is everywhere in Southeast Asia, in Indonesia too. Okay, next please. Yes, could you move to the next? Okay. And in the upland areas, self-help and collaboration of fishermen and agricultural people collaboration is very important. They are collaborating in Tagalog language, bayanihan, a voluntary collaboration, helping each other, voluntary pool of resources. So fishermen are asking agricultural people to help. Agricultural people are helping fishermen, so that natural resources will be both benefited. Okay, that's very, very important and very interesting. In Tagalog language, they call it bayanihan. Okay, there are very similar wording, similar terminology in Indonesia, in Japan too. Okay, voluntary, traditional help. Okay, self-help. Thank you. Next, please. One of the traditional wisdom is utilizing sanctuary systems, designing. In the Philippines, they call it the Yang Kao sanctuary zone. From time to time, in each time they will make, construct such sanctuary system so that they can keep sustainable catch of fish by providing fish corridor systems. Okay, we have analyzed such system in the lake of Laguna Lake. Okay, so that by doing this, people can catch more sustainable fish catch as much as possible for a long time. Okay, next please. And with the help of Department of Fisheries of the Philippine government, is the minister of the fish, can help, and next to him is myself. We are asking the fishermen how to catch fish. Okay. This is me, and this is he's the chair of the fishery department of the Philippines. Okay, next please. And we had a very enjoyable moment of good fish catch by helping each other. In the Philippines, Laguna, in the Philippine Tagalog language, they call it a bayanihan. Gotong loyong in Indonesia, bayanihan, helping each other, is still remaining, surviving in such rural areas. Gotong royong in Indonesian is there. So self-help and voluntary community assistance is very, very important, for maintaining good natural resource management. Thank you. Next, please. Okay, so we need to adopt resource management, not only traditional wisdom, we can adopt more modern and more intelligent ways of resource management with the help of local people. There are two such, new systems which Japan have been doing. One is the PES, Payment for Ecosystem Services. Many types of payments, not monetary, not necessarily the monetary, but also helping each other. Okay. The second is eco-certification, ecological certification by local governments and central governments too. Okay, such new schemes will be helping helping natural resource management, okay, in rural areas and fishery resources, okay? So voluntary and mutual beneficial contracts for sustainable resource use. In Indonesian terminology, gotong royong is very, very important, still there, okay? So traditional wisdom is still surviving, which is community-based and participation system by the local people. Next, please. An alternative way of maintaining sustainable resources is eco-certification. So we have conducted eco-certification research in Japan and also in Lampung, Indonesia for several years. That is the agroforestry-based eco-certification system, and whether PES, payment for such ecosystem services, can be possible or not in Lampung areas, South Sumatra, Indonesia. Okay.
I'm sorry, Prof. Kada.
Yes?
The times is limited.
Oh,
5
okay,
minutes.
sorry.
Make it fast.
Okay.
Sorry.
Okay, so that's the point. So there are in conclusion, let me see. How many more minutes can I talk?
5
1
minutes.
or 2 more minutes?
5 minutes, Prof.
Oh,
5
okay,
minutes.
okay. So,
Sorry.
so PES study, payment, monetary incentives is one of the key elements for local fishermen, local agriculture people to move for sustainable resource management, resource use, okay? That's one of our conclusions. Various types of payments is possible, not necessarily monetary, but also in physical ways as well. Okay. Next slide, please, if there is some more time. Okay. Can you move it next? That's
Next
the
slide,
end,
please.
The end of the story.
Next slide, please.
Yes, please.
Next slide, please.
Okay, another element of important thing is relationship, okay, of between upstream and downstream, because natural resources are related in terms of pollution, in terms of resource use, and those two areas can be related and connected by various types of payments. With the use of ecosystem services, okay? Voluntary and mutual beneficial transactions be possible, okay? And we have been using that system in Japan for many, many years. And it could be adopted as well in other parts of Southeast Asia as well, okay? So payment for ecosystem services can be adopted in terms of agroforestry and downstream resources. That's the point, okay? Okay, maybe time is limited. Okay, I think that's all I wanted to say, okay? Thank you very much for taking this opportunity, okay?
Thank you very much for wonderful presentations, Prof. Kada.
Thank you.
Yeah, the idea of green structure for builds the sustainability, environmental developments. I think it's important for whoever who involved in the agricultural activities. There's one questions here at chat box.
Yes,
Could you
please.
share
Please.
more your experience?
Yes.
There are a question from Rianida Taisa, the students
Okay.
from Polytechnic Negeri Lampung. Could you share
Okay.
more about your experience in other countries, That is how they are improved the quality of water and soil, which already stands by pollutants, sediments like heavy metals. Thank you.
Oh, okay. Well, in order to remove, such heavy metals, we need, there are basically two things. One is to reduce the use of such, heavy metal materials, okay, from industry, from agriculture, and from human activities, okay? How to reduce such, inputs of heavy metals things. And secondly, how to remove heavy metals which have been already contained in the water is another, very important steps of, reducing the damages, okay? In Japan, we have been doing so much inputs, directions, okay? I think Indonesia and the Philippines and other Southeast Asia countries, it is, now is the time of, such steps of how to reduce and remove, heavy metal things and other pollutants from the water, okay? Thank you.
Another questions, probably for
Yes.
the last year from Dayang Berliana. It's a lecturer of Polytechnic Negeri Lampung. How to
Okay.
protect our food security to make sure agri-food system can continue producing despite the world challenge. Science and innovation are vital in ensuring the system for resilience goals. So how can we produce more, healthier and safer while protecting our environment?
Oh, okay. Of course, sustainable agriculture for, for production and consumption of good quality food. So I think producers and consumers in Indonesia, for example, have to collaborate. What kinds of agricultural production should be more enhanced and produced, in order to reduce unnecessary imports, and also how to produce agriculture production without unnecessary inputs. Okay, too much inputs, too much nutrients are also sometimes very dangerous for our health. So we need to improve both the quality and quantity of agriculture production. Okay, that's the answer.
Okay, I think it's great discussions here for the so it's over the session with Professor Kada Sensei. I think, ladies and gentlemen, it is our great opportunity to gather together here, sharing technology and also innovation for better life and better environmental. And then we go to the gifts session for express our gratitude for your join our occasion in second international conference on agriculture and applied science. Please, Bu Fadila Margasati, who is chairperson for ECOAS, to give the certificate for Professor Kada. Please, Bu Fadila.
Thank you.
Okay.
Thank you very much.
Yeah. This is Bu Fadila.
Kada, Prof, thank you for being at ECOAS Polinela. We hope to meet directly with Prof Kada. If you go to Indonesia, don't forget meet me. I'm sorry if it's not a good English, Mr. Prof Kada. Thank you very much. Welcome to Polinela if you go to Indonesia. Thank you.
Thank you very much.
Thank you very much, Bu
hope to visit.
Fadila Margasati. Big applause for Professor Kada Sensei. Hopefully you always healthy and happy. And we hope we could have any opportunity in the next conference to attend Indonesia and visit Lampung, especially for Polytechnic Negeri Lampung. Thank you so much, Prof. Kada.
Thank you very much, Dr. Bye-bye. See you again.
Okay, allow me to go to the next sessions with the fourth presenters. The last but not least, we have Dr. Anung Wahyudi. Dr. Anung Wahyudi. Hello, Dr. Anung. He's from Bandung live because he doesn't.
Good morning, Dr. Fitri.
Okay, good morning, Mas Anung. Okay, Mas Anung, allow me to inform your curriculum vitae first. Anung Wahyudi got a PhD program from Shizuoka University, Japan at 1918 in molecular breedings. She finished the history of the bachelor from Brawijaya University and the magister degree from University of Gajah Mada. And he joined the Peripi Bogor as member of Plants Breeding Associations. Now he's in charge as the head of study programs of seeds technology breeding in Polytechnic Negeri Lampung. Today, he wants to bring the research related with the another lycopene content of the tomato. Am I right, Mas Anung? We usually know lycopene, but now another kind of the proteins is lycopelins, and Mas Anung will be explain more about that. Please, Mas Anung, the time is yours.
Well, thank you very much, Dr. Fitri. Can you hear my voice clear over there?
Yes, very clearly.
Okay, I permit to share my PowerPoint presentation. Okay, thank you very much, and to the Honorable Director of Polytechnic Negeri Lampung, Dr. Sarono, so thank you very much for the opportunity and then time to me, and also thanks to and the Honorable all keynote speakers. So I think they are very senior compared to me, I think. So Professor Bunyamin Taran, so from Canada, and then Professor Rosma Ahmad from Malaysia, and then Kada Sensei, konnichiwa Kada Sensei from Kyoto, Japan, and then thanks also to the chairman of the conference, the second international conference on agriculture and applied science 2021, Mrs. Fadila Margasati, and also all steering committee, and thanks to moderator Dr. Fitri, and thanks to the all participants of this conference. Okay, I would like to present about our research about the lipocalin proteins in tomato. Yes, thank you. I would like to introduce myself. My name is Anung Wahyudi. Actually, Dr. Fitri already introduced me about my formal degree. I graduated from Shizuoka University, Japan, and then a master degree from Gajah Mada University, and then undergraduate from Brawijaya University, and diploma degree from IPB University. And then actually non-degree Genesis program from Shizuoka University and PhD in Gifu University So totally 5 years study PhD in Japan, and then work experience from starting 2012 to present, working on the lecture and researchers at Polytechnic Negeri Lampung. But before that, already worked at the plant breeders at the PT Agrimakmur Pertiwi and PT BC International, and then agronomist at the PT Tanindo Subur Prima. If you're interested with our research, so you can visit our ORCID ID or Scopus ID. And then I would like to introduce our research about lipocalin proteins in tomato. Lipocalins, what is lipocalin proteins? So I think all of you thinking about that lipocalin are one of protein families widely distributed in bacteria and then invertebrate and vertebrate animals. But do you know that very little is known about plant lipocalins? And then plant lipocalins divided into 3 families. The first is temperature-induced lipocalins, divided TIL1 and TIL2, and then chloroplastic lipocalin, or CHL, and then lipocalin-like protein, VDS and ZTP. What the function of this protein in the plant? So this protein actually have function. First is environmental stresses, and then induction of apoptosis, and then membrane formation and fix, regulation of immune response, and then also cell growth and metabolism adjustment. And then to differentiation of lipocalin and tomato plants, so we were performing the following experiments using overexpressed TIL1, TIL2, and CHL, and also gene silencing plants. Actually, Professor Bunyamin Taran, in the first keynote speaker, present about the topic of the CRISPR and then Cas9 about genome editing. So that is the new technique of molecular breeding in Canada, I think in all the world also. But in Indonesia, is still I think not to be familiar because it is new technique. But in here, I would like to present about our study that Dr. Suzuki in 2015 already published on in the PLOS ONE journal that she said proteome data analysis showed that different post-translational modification pattern in the different colors of tomato. As you can see in here, in the red tomato and then black tomato and white beauty, so TIL 2D page, at least the 21 kilodalton and the 19 kilodaltons in the red tomato of microdome. So CHRC also detected and already published it by Dr. Suzuki et al. 2015 also that plastid lipid associated protein in this protein also detected in microtome and also white beauty. And then this is the gene structure of the TIL1, TIL2, and CHL. This is intron and exons. At least 372 until 2050, 79 of intron in the TIL1. And also, we're thinking, how about the phylogenetic tree of these lipocalin proteins, TIL, and how about the phylogenetic tree of CHL also? And we tried to research about this, that the TIL2, at least in here, as you can see, we use the phylogenetic tree in FR. So TIL2 higher similarity with the Nicotiana tabacum or tobacco TILs, and TIL1 in here, as you can see, that higher similarity with potato TILs compared with idle TILs. And then how about the chloroplastic lipocalin? The chloroplastic lipocalin in tomato higher similarity with the Solanum penduli CHL and Solanum tuberosum CHL comparing with others. And then we're thinking again, how about the expression analysis in the 4 developmental state of tomatoes, and then where is the localization of this lipocalin protein in the plant cell? So we use the microtome. This is microtome. I introduced that microtome is dwarf tomato cultivars and then small size and red ripening fruits. and also rapid growing and easy fruit setting. Microtome also can growing better to the seed under fluorescent light and then short life state within 15 until 90 days after sowing. So because of this characteristic, microtome become the model cultivar for functional analysis of lipocalin protein in tomato. And then in here, I would like to introduce about how the expression, relative expression in the 4 developmental states of tomato fruit. So in the tomato fruit, we know that in the green state and then yellow state, orange state, and the red state. In the tomato fruit, when we check the TIL1, TIL2, and CHL, as you can see in here, in the yellow state, a higher content of the chloroplastid lipocalin, and then in leaf, as you can see, TIL1 leaf in the 3-week and 6-week a little bit the same, but in the TIL2, as you can see in here, 3-week and 6-week in the leaf of TIL2 is dramatically decreased in here. And then to check the subcellular localization of this protein in the plant cell, so we use the subcellular localization by GFP fused TILs and CHL proteins. So we make some construct. We use the PUGW5 vectors and then PUGW6 vectors. So this is the genes TIL1, TIL2, and CHL. So we use this construct, and then by gene gun barrels, so PDS 1000. This is DNA coated gold particle, and then we check in the Nicotiana tabacum and Nicotiana bentamiana leaf cells, and then check by confocal laser scanning microscope. And then what the results? As you can see in here that in our data, this is the control. So in case in the Nicotiana tabacum leaf cells, as you can see the TILs, TIL1 and TIL2 in here are localized in the plasma membrane, and then plastid, and then nuclear, and then reticulate structure. CHL or chloroplast lipocalin in tomato localized in the chloroplast. As you can see in here, we use PUGW5 vectors that we check by GFP fuse. The results show that CSL localized in the chloroplast. And then how to understand the function of this lipocalin protein in plants? As you can see that this protein have many functions, but we want to know in the tomato plants. So we make some schematic research that, as you can see in here, we must make first, if we make the overexpression using cauliflower mosaic virus 35 promoter, and then in other side we use the gene silencing using VIGS system. So in here we insert gene to the plant, but in here we make the gene silence in the plant. So a little bit difference. So we make overexpressed plant TIL1, TIL2, and CHL using 35 ICE promoter, and then we make the gene silences plant by PTRV2, PDS, and then PTRV2. This is gene, and we use positive control PTRV2 CHRC. And we already succeeded. This is the method construct. This is our construct. We use PGWB8 vectors. This is nose terminator, and then 35 promoters, and then gene of interest. And then we already success, and then we make, how about the protocol to make the transgenic plant? To make the transgenic plant, we use agrobacterium infection in the cotyledon of microtome, and then we screening by PCR. And this is the results. So we already success make transgenic TIL1, transgenic TIL2, and transgenic CHL. So we already success, and in here we publish in the some journal. And then to understand the function of lipocalin, we also must be understand how about if this protein silence in the plant. So we make the virus-induced gene silencing VIGS to interesting the rapid alternative, I think, this method. So for example, like this. This is target gene, cDNA, and then the binary VIGS vector, and then by agrobacterium inoculation in here, in the cotyledon and in the fruits. How about the metabolism working in here? So single-stranded RNA to ensure the double-stranded RNA. So double-stranded RNA cleavage, this is degradation and then amplification transport, and then we check the silencing phenotype in the plants. So this is the result of RT-PCR. As you can see, we already succeeded making transgenic and succeeded making gene silenced plants. And we check by the, as you know, that the region 1000 base pair upstream of the start codon of this gene, we check by plant care. And this data show that TIL1, TIL2, and CHL have function to the light responsive. So in this gene of this protein, that is correct to have function to the light responsive, and also have function, as you can see in here, about the heat stress responsive also. Heat stress in now I think a big problem to the plant. So global warming like this, so we must be understand to plan to mechanism how to protect this. And then we make some design experiments, so we know the function of this protein. We condition in the light condition, in the dark condition, and then how about the response of this plant. As you can see in here in the germination in dark condition, this is control wild type. As you can see, a transgenic plant is better developing and better growing up and faster, I think, comparing with wild type control in here in the light condition and the dark condition. In the dark condition, tomato hypocotyl is elongation like this, but the transgenic plant already stable like this. And then how about in the 12 days in the treatment by light and the dark condition? As you can see, this is wild type, so control. A transgenic plant is faster to growing. As you can see, this is leaf, and then hypocotyl also, anthocyanin in here also, so it in here. So this result, what the meaning? So this result is provide clues that lipocalin could play roles in the responding to light and reactive oxygen species. I will explain what is the danger of the reactive oxygen species in the plant. This is light condition. We make some experiment in the 405 micromole photosynthetic active radiation, and then in here 405 and here 105. So we check in the wild type plant, and then this is in the transgenic plant. So transgenic plant little bit better for growing and then faster and look like a bullwheat and then curling. But fruits in the transgenic plant, a transgenic plant of TIL1, TIL2, and CHLs, they will make a bigger fruit compared with the wild type. So this is the data. So we check in the tree plants, so plant growing faster, and then also in the fruits, a bigger comparing with wild type like this. And then, as you can see, we check a 40 until 50 fruits also. In the average also, the transgenic plant is look the fruit of tomato is bigger comparing than wild type control. And then we understand again, how about the cytological analysis? So how about the cells, the bigger of fruit, that is what the reason why fruit of tomato can bigger in the transgenic plant. So we make some experiment like this, and then this is the result that cell size of pericarp was expanded in the overexpressed lipocalin fruits. As you can see in here, this is wild type control, and this is transgenic plant. So a little bit in the pericarp of wild type plants, exocarp in here, and mesocarp intercellular space, but didn't find intercellular space in the overexpressed TILs. So this data, I think, interesting because we know that cell expanded, so become widen the fruit a bigger comparing with the wild type. And then in other research, we must be know how about if this protein of this gene is silenced in the plant. So when we check in the silenced in this plant, as you can see in here, wild type can growing like this, but in the silenced plant is very slower to growing. In fruit also look like this. So we check also in the fruit, and you know the result like this. So in the when we check heat stress using 70 degrees Celsius in the 5 days of plants, as you can see the wild type is curling, and then wild type cannot growing good condition here. But in transgenic plant, as you can see, flowering is faster and then plant can growing better. That means TIL1, TIL2, and CSL have function. Hello? I'm still have time, Dr. Fitriani. Hello?
Yes, yes. You still have 5 minutes.
Okay, okay, thank you. I will continue to my presentations. And this is the experiment that this protein have function to the heat stress as you know. And then how about the reactive oxygen species? So we think, and then as I think all of you didn't understand what the problem with reactive oxygen species in the plant. I would like to explain like this. Various experiment stresses lead to the excessive production of reactive oxygen species. This is causing the progressive oxidative damage in the cell death. So if a plant attacked by a ROS, so plant will be dead. So to understand this phenomenon, so we must be understand hydrogen peroxide, and then we check by NBT staining and DAB staining. So this is the result. As you can see that in the light condition, heat condition, and normal condition, and the blue color in here mean blue presence, blue colors means the formation of the superoxide. Superoxide is very dangerous. So as you can see in here, in the wild type superoxide contain a lot of superoxide, but in the transgenic plant is a little bit lower. And then about the hydrogen peroxide also. So wild type plant like this, high content of hydrogen peroxide, but in the transgenic plant is very lower containing comparing with the wild type. And the silences plant, as you can see, the silences plant is contain a lot of superoxide and contain a lot of hydrogen peroxide. So in the silences plants, that means plants didn't have this. So we check also by superoxide dismutase. So superoxide dismutase, as you know, that this is critical antioxidant enzymes that protect organisms from reactive oxygen species caused by adverse condition. So we use SOD1 and then SOD3 and SOD6 to this experiment, and then we check the RT-PCR of the superoxide dismutase in here. As you can see, this content of superoxide, and then this is also the leaf, flower, and fruit we already checked, and then this is content of superoxide here also. And I would like to, my presentation is summary. So I showed to you today that I would like to make some summary. I hope all of you can easily understand what this presentation. Like this. Stress, so plant have stress from light and heat stress or oxidative stress. This is problem. If plant cannot protect this, plant will be dead. So we use transgenic plant, the TIL1, TIL2 gene insert in the plant. When plant contain that inserted this protein, so plant will be, first as protein is